Hello, everyone, and welcome to today's webcast on new approaches in bispecific antibody development. I'm Tim Wright, editor of Contract Pharma, and our webcast today is being sponsored by Samsung Biologics. They're a fully integrated CDMO offering state-of-the-art contract development and manufacturing services. With proven regulatory approvals, the largest capacity, and fastest throughput, Samsung Biologics is an award-winning partner of choice and is uniquely able to support the development and manufacturing of biologics products at every stage of the process, while meeting evolving needs of biopharma companies worldwide. For more information on them, visit samsungbiologics.com. And our speaker today is Jina Kim. She's a senior scientist at the Bio R&D Center at Samsung Biologics. Jina studied oncology at the Department of Medical Science at Yonsei University in Korea. And at Samsung Biologics, her work focuses on the development of bispecific antibodies and antibody engineering. Today, Jina is going to discuss new approaches in bispecific antibody development and introduce the SQL Bispecific Antibody platform that enables high purity, thermal stability, and manufacturability by eliminating mispairing risks while maintaining high quality structure for your novel findings. We will be accepting questions throughout the talk. You can send those in using the box at the bottom of your screen, and questions will be answered after the presentation in a brief Q&A. If you're experiencing any technical difficulties, you can type those into the question box as well, and someone on the back end here will help you out. At this point, I will turn it over to Jina.
Good morning, everyone. I'm here to talk to you about new approaches in bispecific antibody development today. In the first, I'll briefly introduce about bispecific antibodies development. In 1960s, for the first time, the bispecific antibody concept was demonstrated by Dr. Nielsen. After 30 years, bispecific antibodies has developed intensively after novel technology was turned up by Genentech. Since then, a wide variety of bispecific antibody platforms have been developed, and more than 20 types of commercial technologies have been introduced so far, and 7 therapeutic bispecific antibodies have been approved. The global bispecific antibody market is expected to grow 5-fold in 6 years, from $4 billion in 2021 to $19 billion in 2027. And also the CAGR is to be increased 30% during the same period. This market growth is driven by the growing pipelines due to the advantages of bispecific over mAb. There are over 600 bispecific antibodies in clinical activity at the moment. Furthermore, we expect that the development of bispecific antibodies will be accelerated even more. This slide shows our manufacturing facilities in main site of South Korea. Samsung Biologics was established in 2011 and began GMP production in 2013 in a total capacity of 30 kiloliters. Now we have 420 kiloliter manufacturing capacity, and we will continue to expand manufacturing facilities to 604 kiloliter until next year. This slide goes through the history of the Samsung Biologics about CDO business. In 2018, Samsung Biologics expanded our capability into the CDO business for increasing demand of early phase drug development. And in 2020, we launched our additional facility, that is R&D Center in San Francisco, where online development service can be provided. More recently, we have launched our proprietary bispecific antibody platform, SQL, and Developability Assessment platform, Developing. We invest to promote collaboration with our clients has led to over 90 projects in 4 years. And also, our proprietary technology increases success of clients' pipeline. Moreover, we provide seamless tech transfer to Samsung Biologics' high-capacity CMO facilities when pursuing large-scale manufacturing. This slide gives an overview of the broad range of molecules that we worked on in CDO business. We have experience in developing various molecules, including bispecific antibodies. Looking at the titers in our project, we have achieved the titer for bispecific to be in around 4.8 grams per liter and bispecific active fusion protein to be in 3.6 grams per liter. As you know, the bispecific antibodies have pros and cons. The advantage of bispecific antibodies is that they can be applied to various targets by combining two antigens at the same time, and also increase therapeutic window compared to the combination therapy. On the other hand, there are still problems such as incorrect pairing issues, low purity, and low manufacturability. If there is no antibody engineering, the theoretical correct pairing yield at 12.5%. It is quite low. So we want to overcome these limitations in our S0 bispecific antibody platform. Next, I will introduce a new approach to the bispecific antibody platform. S0 is Samsung Biologics' proprietary bispecific antibody platform. It has several distinct characters. The platform is a symmetric and IgG-like structure. As you know, Nobin-Hall technology has made great contribution to solving the heavy chain mispairing. And there are also many solutions for avoiding heavy chain light chain mispairing, such as making charge difference, cross-cell location, and common light chain. We want to avoid not only heavy chain mispairing, but also heavy chain light chain mispairing. So we added two Nobin-Hall structures within one subarm and FG region of antibody. These two added and optimized Nobin-Hall structure used to avoid heavy chain mispairing and heavy chain light chain mispairing. In addition, the asymmetrical structure is advantageous for single-cell production because it is easy to distinguish initial impurities due to the size differences. And also, it has easy in-process control during manufacturing as well. And all composed domains derived from human origin. It can be minimized immunogenicity risk. Here is a case study using S-dual bispecific antibody platform. We tried to produce different types of bispecific antibodies with two different methods to our platform. In first case, VEGF and HER2 bispecific antibody, it was combined two antibody sequences derived from different monoclonal antibodies. And in second case of HER2 and HER3 bispecific antibody, the sequence was derived from one bispecific antibody. The following slides are about the physical-chemical properties and in vitro in vivo efficacy study of our bispecific antibody. After transient expression with both molecules, we conducted three-step purification such as protein A, ion exchange, and Higg. In site exclusion analysis, each molecule is 98 and 99% monomer purity are confirmed respectively. When we analyzed the previous samples in capillary electrophoresis, we confirmed that the correct heterodimer was formed greater than 96% under non-releasing conditions respectively. This purity data shows that we overcome not only heavy chain mispairing, but also heavy chain light chain mispairing problem of existing bispecific antibodies through S0 bispecific antibody platform. And also, when we tested thermal stability of both molecules with DSC, T onset is 53 and 58, TM1 is 63 and 68, TM2 is 73 and 86. These temperatures show comparable values to conventional mass. This slide shows in vitro efficacy study in VEGF and HER2 and bispecific antibody. When we tested ELISA for binding affinity of VEGF and HER2 bispecific antibody with parental map. Those antibodies show similar binding affinity to the VEGF and HER2 single antigen respectively. And looking at the upper right graph, when VEGF and HER2 dual binding was measured, our bispecific antibody can bind to both sides of them at the same time. In the study of HER2 and HER3 bispecific antibody, there are our S-dual and reference bispecific antibodies. The binding affinity of both molecules were measured by SPR method. We immobilized the capture antibody against bispecific antibody and checked the KD value for each single antigen. The KD value of each antibody was at around 1 nanomole for the HER2 antigen, and the KD value for the HER3 antigen was at around 20 nanomoles. The results are similar between our bispecific antibody and reference molecule. In order to check the dual binding affinity using the same antibodies, we fixed both bispecific antibodies and confirmed the EC50 value for the HER2 and the HER3 antigen. The results of this study also show similar values within the error range between our bispecific antibody and reference molecule. This slide shows the fixed binding assay and the proliferation assay results to confirm the efficacy of bispecific antibodies. Those assays present similar results and IC50 value between our bispecific antibody versus the reference molecule. In vitro assay results of the bispecific antibodies suggest that SQL Platform has comparable efficacy with reference molecule. Next slide, when we tested in vivo xenograft mouse model with our HER2-HER3 bispecific antibody and reference molecule in the same design of their preclinical studies, we observed that both bispecific antibodies show better growth inhibition to HER2-positive cell line than trastuzumab and pertuzumab combination. And also, SQL and reference bispecific antibodies show similar survival rate in this in vivo study. Through this data, we confirmed that our bispecific antibody platform has comparable efficacy to their parental bispecific antibody. When we track the immunogenicity of our bispecific antibody platform using the in silico prediction method, it shows similar level of immunogenicity as other therapeutic antibodies. As you can see in the left graph, when we compare to other therapeutic antibodies, it was confirmed that the level of immunogenicity is similar to other humanized mAbs. And at the right figure, when comparing the high score of immunogenic position of two other antibody drugs, it has lower score than other antibodies. Nevertheless, in order to confirm more detailed immunogenicity data on our bispecific antibody platform, we plan to study an additional immunogenicity through PBMC-derived T-cell assay. So now I'll introduce our another proprietary platform, the Developability, which is for Developability Assessment Tool. Many pharmaceutical companies are looking for candidates that have pharmacological effect and potential to be novel drugs. Among many biology molecules, it is important to exclude the candidate which has poor stability and developability. Our Developability platform can rapidly evaluate pharma stability and chemical stability for the potential candidate in early phase. This prediction is more effective to be performed before the initiation of cell line development. Especially, the Developability platform makes synergy with SQL Platform. When the client provides their antibody sequence, it can be applied to the SQL bispecific antibody platform to produce with transient expression method for small-scale antibody production. If client wants to add it can be produced for supplemental study. And then we evaluate concentration, thermal stability, aggregation, and purity profile of the product. After having analytical results, clients can make a decision for going on the process development or not. This slide explains the analytical results, including SQL Platform with Developability. When we compare the SQL Platform with other monoclonal antibody and novel-based FTG fusion proteins, SQL Platform has similar thermal stability to monoclonal antibody and good stability at low pH. These results indicated that directly links to good developability of our bispecific antibody platform for further development. Our Developability platform provides an accurate evaluation of initial candidates that makes to minimize the potential risks such as low stability and aggregation. And in the development process, Developability also can reduce the uncertainty, time, and cost. Through this Developability Assessment tool, the success rate can be increased in developing new pipelines. In summary, Samsung Biologics has accumulated extensive experience in the development of not only monoclonal antibodies, but also bispecific antibodies. Our novel proprietary bispecific antibody platform, SQL, enables robust heavy chain to light chain pairing to ensure high purity. And it also presents high thermal stability and low immunogenicity. Our platform preserves binding affinity and potency, which are comparable to those of their parental antibodies. Notably, our SQL platform provides high quality and good manufacturability without any loss of function. Moving on to my last slide. Now we have two R&D centers for CDO services in Korea and San Francisco. In San Francisco, R&D center where cell line development service can be provided. And also, we can provide a seamless one-stop solution from cell line development, DS, DP, and fill and finish in main facility of South Korea. The SQL development and developability assessment services can be provided at both sites. This is my last talk, and if you have any questions, I'd be pleased to answer them. Thank you for your attention.
Okay, great. Thank you so much for a really informative talk there, Jina. As she mentioned, we do have some time to answer some of your questions. So if you did have a question and weren't able to get it in during the talk, now's the time you want to go ahead and type it into the box at the bottom of your screen. Okay, so first question is a two-part question. This attendee would like to know what is the ratio of heterodimerization from culture media, and secondly, what kinds of impurity can be seen?
There are a few hash molecules could be formed at very low rate, and they can be removed via purification steps. Samsung Biologics will develop the line by optimizing ratio to minimize impurity.
Okay, thank you. Next, this attendee asks, which KIH technology did Samsung Biologics use, and which amino acids were changed?
The SQL platform uses typical novel sequences such as 366 threonine, 368 leucine, 407 tyrosine.
All right, great. Thank you. Moving along, are FCCH3 dimer and SABCH3 dimer the same?
The same novel sequences is applied, but the presence or absence of disulfide bonding is different.
Okay, thank you. And we're going to take one more question before wrapping things up today. This attendee would like to know, in general, what is the contract procedure like if they're interested in partnering with Samsung Biologics?
We offer our SQL service by first entering into MTA with our clients. The final deliverables are the molecule production and the report of its physicochemical properties. If the client is satisfied with the material and its physicochemical properties and wishes to proceed to stable cell line development, the client needs to enter into the commercial license agreement with us. The benefit of initiation your bispecific antibody development with SQL is that it has favorable contract terms with competitive pricing.
Okay, great. Thank you, Gina. Okay, so that's going to bring us to the conclusion of today's session. If we didn't get to your question, Gina has your contact details and she will follow up with you directly. So thanks again for a terrific talk. It's really informative, and thank you to Samsung Biologics for today's sponsorship. I'm going to be sure to visit them online at samsungbiologics.com. And thank you to everyone in the audience for your attendance and participation today. This session will be archived for 1 year on our website, contractpharma.com, and you'll be receiving an email from us shortly when the session is available for on-demand viewing. Thanks very much for joining us, everybody, and have a great rest of your day. Take care.